Hi, everyone. I hope all of you are safe and well. I'm very excited to share my work here at OHBM. So as we all know, the global fMRI signal refers to the mean time series average across all voxels in the brain and has been associated with physiological processes. For instance, here we see that regions strongly correlated with the global signal, such as visual and sensory motor brain regions, overlap with regions associated with fluctuations in carbon dioxide. Carbon dioxide is a potent vasodilator, which means that fluctuations in carbon dioxide change the vessel diameter, bearing cerebral blood flow and thus bold signals. Carbon dioxide fluctuations are usually captured indirectly using a respiratory belt that measures the breathing rate and depth. Also, changes in heart rate are associated with global signal fluctuations, presumably through changes in cerebral blood flow as well. So given all these, global signal regression, or GSR, is a popular preprocessing method to account for physiological processes. Further evidence of the benefits of performing GSR was provided recently by Lee and colleagues, where they showed that GSR strengthens the association between resting state functional connectivity and behavior. So before diving in on the controversy surrounding GSR and our simultaneous EEG fMRI study, I would just like to provide more evidence from our lab regarding its utility. To better understand the effects of GSR, we investigated the effect of physiological processes on estimates of functional connectivity when these fluctuations are not removed from the fMRI data. And the results of this I'm presenting in more detail at poster 1000. So briefly, we modeled low-frequency physiological fluctuations in heart rate and breathing signals using model-based techniques and external physiological recordings. The main idea is simple. Heart rate and breathing signals are convolved with their respective physiological response functions and added together in order to yield a time series that reflects systemic low-frequency oscillations, or SLFOs. Then we used a framework that allowed us to quantify the extent to which functional connections were affected by SLFOs at the subject level. Then we averaged these connectome patterns associated to SLFOs across a large number of subjects from the Human Connectome Project dataset. And we found that SLFOs exhibit systematic biases in estimates of functional connectivity. More specifically, they induced increased connectivity within and between the visual network mainly. And in these violin plots, we present the similarity of the connectome patterns related to SLFOs with the connectome patterns obtained after preprocessing the data with several denoising strategies. This similarity was computed at the subject level and reflects the extent to which the connectome profile is confounded by low-frequency physiological artifacts. The top row corresponds to the raw data, and the subsequent rows reflect preprocessing strategies ordered from mild to more aggressive pipelines. And the green color corresponds to the same pipelines with the addition of global signal regression. So if we look at the top row, we can observe that SLFOs severely confound the connectome patterns. And with regards to the preprocessing strategies, I won't go into much detail, but I would just like to point out that FIX, the chosen preprocessing pipeline for the HCP data, failed to remove this bias, which is consistent with recent studies. And GSR seemed to be the only method that successfully removed the effects of SLFOs. If you want to know more about the methodology and the effects of other nuisance processes and their subject discriminability, please check out our preprint or our poster. And well, actually, hopefully, I will have talked to many of you about this project by the time you listen to this presentation. And so now, given all this evidence, why is GSR so controversial? Well, it boils down to the fact that it's not clear whether it also removes neural activity. For instance, the global signal amplitude has been associated to EEG alpha power, and also fMRI global signal transients seem to co-occur with the stereotyped spectral patterns in LFPs. Finally, a recent study also showed that individual differences in the global signal topography seem to be related to behavior and cognition. So given all these, the goals of this study were to characterize the processes underpinning the global fMRI signal as well as to examine the effect of GSR on fMRI connectivity patterns associated with physiological and neural fluctuations. And in order to achieve this, we collected simultaneous EEG fMRI data, as well as physiological signals, specifically pulse oximeter and breathing valve signal, from 12 young subjects from 2 resting state scans that had a 15 minutes duration each. The fMRI data was preprocessed using 10 principal components from white matter, and the EEG power bands at delta, theta, alpha, and beta bands were extracted, averaging this power time series across all 64 EEG channels. So first, for each scan, we computed the partial correlation of SLFOs and the global signal, which is shown here in orange across subjects, as well as the correlation, the partial correlation of EEG power bands and the global signal, which is shown in green across subjects. And as we can see, SLFOs explain a large variance in the global signal, whereas, for example, alpha power exhibited only a weak association with the global signal. And to be more specific, we observed a negative correlation between alpha power and the global signal. Regarding the other EEG bands, we saw that none of them showed a significant contribution to the global signal when we compared it to surrogate data. We then used the same framework as in the previous study to obtain the connectome patterns associated with SLFOs and alpha activity. For SLFOs, they demonstrated similar patterns with the ones observed earlier with the ACP data set. So namely, mostly the visual connection were the ones that were mostly affected by SLFOs. And for alpha power, we saw that it was linked to high correlations within the visual, dorsal attention, and the fold mode networks, as well as negative correlations between the DMN and the visual and dorsal attention networks, which is consistent with the literature. And we then computed the similarity between SLFOs and alpha connectome patterns and the fMRI connectome profile at each preprocessing stage. With respect to SLFOs, this similarity reflects the extent to which the connectome profile is confounded by SLFOs at each preprocessing stage. First of all, we see that the preprocessing strategy used, which consists of removing 10 white matter components, achieves a significant reduction in the effects of SLFOs. That said, global signal regression yielded an additional reduction in the similarity between the connectome pattern related to SLFOs with the connectome profile of the fMRI preprocesses data, which indicates that GSR is beneficial even for relatively aggressive pipelines to effectively remove the stereotype connectome pattern induced by SLFOs. With respect to alpha power, removing 10 white matter components enhanced the similarity between the connectome pattern associated with alpha power fluctuations and the fMRI connectome pattern. And importantly, we found that adding GSR to the preprocessing pipeline did not significantly alter the patterns in functional connectivity associated to EEG activity. So overall, what we saw was that the global signal is strongly associated with physiological processes and only weakly associated with alpha power fluctuations. We also show that GSR effectively removes the connectome patterns induced by physiological processes, and it did not alter the patterns in functional connectivity associated with alpha activity. So our results provide evidence that in the context of connectivity analysis, GSR improves the denoising of fMRI data while preserving the signal of interest. So thank you all for listening, and I would like to thank my supervisors as well as all my lab mates and the funding agencies.
Thank you everybody for coming to this talk. The title of this talk is Gastro-Brain Coupling Predominates in Primary and Association Sensorimotor Regions. We're going to work out the meaning of this title together. So when we think about the origins and functions of intrinsic brain activity, the fact that the brain is always active, we tend to associate it with perception of the external world or production of intrinsic cognition like mind wanderings. But what about the fact the brain is always monitoring the internal milieu, homeostasis, in order to keep the body alive? So visceral signals like the stomach or the heart are rhythmic, and in animals at least, they target cortical, subcortical, and neuromodulatory structure. We're going to come back to this image before at the discussion, but we previously showed that the slow electric rhythm of the stomach, which is produced every 20 seconds, is coupled with resting state brain activity in a gastric network. So here I'm going to show a follow-up on this work in which we use a larger sample size and we redefine the anatomy of this gastric network, specifically which cortical regions are coupled to the rhythm of the stomach. So this gastric rhythm is produced in the pacemaker region and propagates to the stomach, and it serves to pace the contractions that are needed to grind and digest the food. But it's present even in the absence of contractions or even when the stomach is disconnected from the brain. It's actually produced by this interstitial cell of Cajal in blue that always produces rhythm and makes synapses with vagal sensory neurons which send this gastric information to the brain, and could potentially also be sensed by the spine, which innervates the smooth muscle. The stomach, this gastric rhythm can be measured using the electrogastrogram, which are cutaneous electrodes on the abdomen, and we can recover the classical peak at 0.05 Hz that characterize this rhythm. And we can easily distinguish the gastric cycles from the faster respiratory or heartbeat activity, which have a much higher frequency range. And you can read much more about this method in our new paper in psychophysiology, where we describe the methods and analysis of this technique. So what we did for this experiment is that we recorded 15 minutes of resting state both fMRI in 63 participants that were fasting for 2 hours, meaning that they were not hungry, not full. And we quantified phase coupling in all the brain's voxels using phase locking value. So this phase locking value doesn't tell us anything about directionality and doesn't take into account amplitude. It will just tell us if the two-time series are coupled or not. And we can see an example here with this orange voxel that is tightly following with a constant delay the stomach in blue. And in gray, we have the example of a non-coupled voxel in which the time delays of the two signals are random. We quantify this using this equation that is basically the vector length of the phase difference between the two signals, and we obtain for each subset a PLV map, a coupling map, and we make a change map using the ECG of other participants. And then we make a t-test and use a clustering procedure to correct for multiple comparisons. We then, once we have the significant map, we characterize the anatomy and the effect sizes of this gastric network across resistant networks, cortical regions, and gradients of cortical organization. So what I'm showing you now is Yeo's 7 resistant network parcellation of the cortex. And now I'll show you the effect size of the difference between chance and empirical coupling with the stomach. And you can see that in somatomotor, here in blue, and in visual resistant networks, we have a lot of overlap with the gastric network. So we quantify this by measuring how the gastric network was represented in the different resistant networks. Around 70% of the gastric network is in this sensory motor region. We also have subcortical structures like the thalamus, the striatum, and the cerebellum. We can say that coupling occurs mostly in sensory motor regions. Then we wanted to have a look at which cortical regions have the largest effect sizes, and for this we use the Glaser parcellation, which use task fMRI, myelin, and connectivity to separate the cortex in 360 regions. And here I'm showing you Cohen Z, a measure of effect size between chance and empirical PMV. And 0.8 will be a large effect size and 0.5 will be a medium effect size. And the regions with the largest effect size are actually in the primary somatomotor cortex. Here I'm showing you a lateral view of the brain, inflated brain, and we can see the primary motor and primary somatosensory cortex, the regions with the largest coupling with the stomach. And we also have large coupling in premotor regions and in cingulate motor regions. So it seems that regions sensing the skin and controlling muscles are more strongly coupled to the rhythm of the stomach. Here I'm showing you a lateral view of the operculum of the in an inflated brain, and we can see that primary auditory and auditory association regions also have large effect sizes, as well as insula granular and mid-insular cortex and secondary somatosensory cortex. But note that the insula is only one of the nodes of this gastric network. We also have coupling in straight and extra straight visual cortex in this view of the posterior part of the brain, which also have large effect sizes, and in the retrosplenial cortex, meaning that non-sensory regions have large effect sizes as well. And note how the gastric network spares the ventral precuneus, that is the core node of the default network. So the gastric network includes large portion of the visual cortex and specific transmodal regions. We then wanted to look how the gastric network is represented in the cortical gradients of the brain, the fact that brain structure and functions varies in a systematic fashion across the cortical hierarchy. So this comes back to the view of Mesulami, which in the periphery of the cortical hierarchy, we have sensory regions like the visual cortex or the auditory cortex. And in the center of the hierarchy, we have transmodal regions like the default network. And this is what Margulies and colleagues did, is that they applied spectral embedding, which is a dimensionality reduction technique, to whole brain functional connectivity patterns, revealing gradients of shared covariance in functional connectivity. The first of the gradients, which explains most of the variance in connectivity, distinguishes between transmodal regions in hot colors and unimodal regions in cold colors, which is default and sensory motor regions. The second gradient actually distinguishes between different sensory motor modalities. And what I'm showing you here is on the y-axis, the first axis that goes from unimodal to transmodal, and in the x-axis, the second gradient that goes from visual to somatomotor and auditory regions. And you can see that for the first gradient, we have a large peak we have a lot of voxels that are transmodal or unimodal. But now if you only look at the gastric network, we can see that there seems to be a bias in the sense that we don't have as many transmodal regions in the first gradient nor in the second gradient, but we do have a lot of sensory motor regions. And we verified that this pattern could not be obtained by chance if the gastric network was randomly distributed across the cortex. So the gastric rhythm recruits preferentially unimodal sensory motor regions and spares transmodal regions. We can now come back to this working model of visceral signals, which I showed you before, which is based on the animal literature and not necessarily on the stomach. It's based on different organs of the body. So a reminder that the PLV doesn't tell us anything about directionality. So a priori, the gastric rhythm, this gastric network could represent ascending, descending, or both ascending and descending influences. So animal models are very important to find out how is the directionality of these interactions. It's very interesting that we can recover most of the hypothetical cortical targets of visceral signals with the stomach in humans, which is quite reassuring. And it's also very likely, we also don't find the intromedial prefrontal cortex, which may indicate that the parasympathetic system might not play such an active role. We think that the spinothalamic tract is likely to play an important role. The spinothalamic tract bypasses the brainstem and sends directly spinal input from the spine to the thalamus and targets cingulate motor, primary and secondary somatosensory and insular granular regions, the same that we found for the stomach. But we have to update our model, our working models, with the new information that we found, in the sense that we found that all the sensory modalities are coupled with the stomach, including audition. How come that all the sensory modalities are coupled with the stomach? I think one possibility is related to the parabrachial thalamic pathway. The parabrachial nucleus is a visual relay and sends massive inputs to the visual thalamus, in the sense that 1/3 of the inputs of the lateral genicular nucleus come from the parabrachial, 1/3 from the retina, and 1/3 amplified by connection from the occipital cortex. We don't know why the occipital cortex is receiving visual information. I have my own hypothesis. I will be very happy to discuss this in the discussion. But just to say that when the stomach is artificially inflated with a balloon, an fMRI measure, we'll see massive deactivations of the visual cortex. So there is something related to sensing the internal wall versus sensing the external wall, in my opinion. We also have probably the influence of neuromodulators, in the sense that serotonin, dopamine, and noradrenaline are targeted by visceral signals. And we probably have the influence of descending pathways away, in the sense that it was recently discovered that the rat stomach, sympathetic control of the rat stomach is controlled by M1, S1, and premotor regions. So it's likely that this gastric coupling is also reflecting sympathetic control of the stomach. And BMPFC and anterior insula is parasympathetic coupling. So, gastric coupling likely reflects multiple ascending pathways and sympathetic control of the stomach. Why sympathetic and not parasympathetic? Well, I think it's because subjects are not doing anything special. They are just laying there. They are not hungry. They are not full. They are just laying there. It may change if they are given some food cue. And thank you very much for your attention. We come back to the title, gastric brain coupling predominates in primary and association motor cortex. And if you have more questions, please come to my poster 1330. Thank you very much.
Okay, so I'm Yameng. I'm from Penn State University. Today, I'm going to talk about the consistent global propagations across cortical hierarchy in the electrophysiological and fMRI signals. The spatial temporal patterns have been found by using a template refining approach to extract the repeated quasi-periodic patterns. Another study also found the systematic temporal lags at different brain regions. Again, decomposition of these lag structures generates multiple lag stress, but these spatial temporal structures doesn't represent the propagation in the fMRI signals. Another study also found a time lag between the sensory regions, such as the visual cortex and the sensory motor cortex, and the anterior cingulate cortex, which is part of the deformal network. So this indicates that there might be a time lag along the cortical axis. Another study decomposing the resting state function MRI connectome, generating a function connectivity map, which is the principal gradient, showed a gradual transition from the primary sensory motor regions to the deformal network. So combining these two, we hypothesize that there might be a propagation structures along a hierarchy axis in the fMRI signals. So to test whether there is a propagation structure along the cortical hierarchy, we project the fMRI signals along the principal gradient, which has a hierarchy contrast. So specifically at each single time point, we sort out of the cortical vertices based on their values on the principal gradient. And as a result, we can get a vector along the cortical hierarchy starting from the low order regions to the high order regions. We then apply this method across all the time points, generating a time position plot, and the vertical axis represents the different hierarchical levels of different brain regions, and the horizontal axis represents different time points. And we can see there are a lot of continuous active band in this time position plot, and they are tilted and traveling from the low order regions to the high order regions. If we take a closer look at this continuous band, we can see that there is a systematic delay of the local peaks along the hierarchy axis, and the fMRI signal of this segment on the brain surface showed a continuous movement from the sensory and motor regions to the deformal network regions. And this indicated that there is some propagation structures along the cortical hierarchy. We observed in the previous slides that there are delays of the local peaks along the hierarchy axis. So next, we developed a data-driven method to map the local peak traveling trajectory without projecting the fMRI signals on any template. So first, we cut the signals into temporal segments based on the troughs of the global signal. So next, we calculate the delay of the local peaks within each segment, which is composed of the delay profile. Then we contact all of the delay profile together to generate a delay matrix. We then use singular value decomposition on this delay matrix. So the principal delay profile, which is the first component of the singular value decomposition, and we can see there is a gradual transition along the cortical axis, starting from the motor and the sensory regions to the deformal network regions. And it also showed a high similarity with the principal gradient. And on both of them, we can see that the first visual cortex violates the cross hierarchy arrangements. The propagation structures are expected to synchronize fMRI signals and affect the correlations in a systematic way along the cross hierarchy axis. And we guess that's why we can see that the decomposing the function connectome can generate a principal gradient, which is along the cortical hierarchy. So to detect the single propagating structures, we use the similar method as before, and which is to project the fMRI signals along the principal delay profile. And we indeed observed that there are some segments which showed a continuous active band along the hierarchy axis. We then used the correlation between the timing and between the delay of the peaks and the hierarchical level to quantify the existence of single propagating structures. We then calculated the correlation of the segments across all the subjects along the principal delay profile. We can see then the correlation distribution along the principal delay profile showed a bimodal distribution and have more positive correlations compared to the control directions, such as the anterior to posterior direction. So this indicates that first, there are significant propagations along the principal delay profile, and second, there are more sensory motor to deformal network propagations indicated by the more positive correlations. So far, we showed that there are propagation structures along the cortical hierarchy in the fMRI signal, but there are two problems. First, the propagation might be induced by the hemodynamic response of different brain regions. And second, the recent studies have found a systematic low-frequency oscillation of blood signals can induce systematic fMRI delays, and such fMRI delays are consistent with blood transit time. So it suggested a hemodynamic response might contribute to the propagation structures. So to solve this problem, we used the electrophysiological signal, which is free of hemodynamic confounding, to see whether we can see the same propagating structures on the electrophysiological signals. We used a dataset from a Japan group. The ECOG signals is recorded under three different conditions: the sleep condition, eye closed condition, and eye open condition. On each monkey, there are 128 electrodes put on the monkey's brain. The principal delay profiles under the eye closed condition in the gamma band frequency showed a cross hierarchy contrast. The correlation distribution along the principal delay profile also showed a heavy-tailed distribution compared to the control direction, indicating there are significant propagation structures along the principal delay profile. We also take a look at the principal delay profiles in the other frequency band and found that the cross-hierarchy propagation only exists in the gamma band power. The previous studies also found that the brain spatial temporal patterns are sensitive to the brain state. They found that the brain spatial temporal patterns are different under awake and sleep state, and the brain spatial temporal patterns are even showed opposite direction under awake and anesthesia state. So we also found two types of propagations with opposite direction. To test whether they are also dependent on the vigilance state, for the fMRI sessions, we divided all of them based on their vigilance level to three groups. We found that under sleepy state, there are more sensory motor to deformal network propagations and less deformal network to sensory motor propagations. We also found the same trend under the fMRI sessions, where subjects were noted to be sleeping, and also observed the same trend in the monkey ECOG dataset. So to summarize, first we found that the cross-hierarchy propagations might result in a principal gradient of the resting state fMRI connectivity. We also found similar cross-hierarchy propagations in the human resting state fMRI and the ECOG signals. First, we found that both of them showed a similar cross-hierarchy trajectory. Second, both of them showed a dominant sensory motor to deformal network propagations. Third, both of them showed dependent on the vigilance state. Next, we also found the ECOG propagations are only present at the gamma band power, and we also found the neural origin of the resting state fMRI propagating activity. For the method part, we developed a new delay profile decomposition to extract and quantify the propagating structures. It can extract and quantify the single instance of propagating activity and have more sensitivity than the session-based method. I want to thank my lab and the funding resources. If you have any more questions, please come to see our poster. And our lab is also opening for the postdoc and the graduate students. If you are interested, please contact Dr. Xiao Liu. And thank you.
Hello, everyone. Thank you for attending my talk today. I'll be discussing global signal topography changes across the lifespan. And as we're all familiar, the global signal is typically the average time series of all brain voxels, and it has both non-neuronal and neuronal influences. So some well-known non-neuronal influences are, of course, respiration and cardiac events, and also head motion. So as Jonathan Power has shown, increases in head motion measured by frame-wise displacement here are positively related to this DVARS measure, which is average whole brain intensity of the global signal. And you can see the same effect for respiration and cardiac events also. So previous research has also shown that there are neuronal influences of the global signal. So in macaque monkeys, Turchi et al. Has shown that if you inject a GABA agonist into the basal forebrain, essentially what you see here on the top here is that with no agonist, the global signal explains a large amount of variance in the overall topography of the brain here. However, if you inject this GABA agonist into the left hemisphere, you see that the global signal, the contribution of it to the variance explained goes down dramatically. And you see the same thing here in the right hemisphere if you inject this GABA agonist to the basal forebrain, demonstrating that there is some neuronal influences of the global signal. So this makes it a controversial issue in the literature. Should we or should we not regress out the global signal? So on one hand, regressing out the global signal will help to eliminate noise. However, on the other hand, you may be regressing out important neural signal. So we sought to further look into this on the neural signal side to see if the global signal topography can characterize differences in brain function and perhaps provide more evidence for this neural basis of the global signal. So the global signal topography is basically you're acquiring the global signal and then you're going to regress that or correlate that with each voxel in the brain. And it's going to show you spatially which brain areas are maximally associated with the global signal. And what you see in typical individuals here is that the global signal is usually maximally associated with sensory motor areas and also visual areas here, as we see in Liu et al. And also in Yang et al. So global signal topography has also been shown to have spatial differences in clinical conditions. So Yang et al. Showed in schizophrenia that the global signal has stronger associations with these lateral frontal parietal areas or the central executive network, and it has weaker associations with sensory motor areas and visual areas here compared to typical individuals. Zhang et al. Looked at differences in global signal topography in bipolar disorder. So they scanned in the individuals with bipolar disorder when they're in a depressive state, manic state, or in a state of euthymia, which is a normal mood state. And in depression, they saw that the global signal had increased associations with areas like the hippocampus. And during manic conditions, they found that the global signal had increased associations with the sensory motor areas. And then in a euthymia state, they found that the global signal had decreased associations in the medial prefrontal cortex. So all in all, demonstrating that the global signal can characterize differences in clinical conditions. So we were interested in our previous research to look at the relationship between global signal topography and behavior, and we built this study, or we did our study following on the framework that Smith et al. Provided, where they did a canonical correlation analysis looking at the relationship between brain and behavior in HCP data. And basically what they found is that a number of connections in the brain here were related to this positive negative axis of behavioral traits or life traits. Essentially, they found that these connections here were positively related to things like life satisfaction, fluid intelligence, sustained attention, financial income, and they found that they were negatively related to negative life traits such as smoking, THC use, drug or alcohol problems, essentially showing the relationship between functional connections in the brain and life traits. So we started to build off the study, and we were interested in what the relationship was between global signal topography and behavior. So we used the HCP data, just like Steve Smith did. We had 1,000 subjects here, 4 runs each, 4 resting state runs. We calculated the spatial topography of the global signal, as I mentioned previously, by regressing the global signal onto each of the vertices of this surface-based data. And we reproduced this typical global signal spatial topography that you see. So the global signal was most strongly associated with sensory motor areas, visual areas, cingulate, some areas in the insula. Doing the principal component analysis, we saw that the most variance explained across this global signal topography was in areas related to the lateral frontal parietal network, so the central executive network here, also in the default mode area. And then the sensory motor spatial layout actually explained the least amount of variance out of these three components. So we then did a canonical correlation analysis, as in Smith et al., where we took these whole brain voxel-wise global signal beta maps, and we put that into a CCA along with 177 behavioral categories, just as Steve Smith did. And we found also a single canonical variate pair where global signal weights here in the lateral frontal parietal network, or the central executive network, was related to this positive negative axis, just as Steve Smith found. So the brain areas here were related in a positive manner to things like life satisfaction, years of education, and they were negatively related to things like aggressive behavior, antisocial personality, THC. So essentially showing that the global signal topography does have an association with behavior and life traits. So building on this, we also were interested in, does this topography change across the lifespan? So of course, this has implications for developmental studies who regress out the global signal. So it may be the case that global signal regression is differentially influencing the data according to what developmental period you're looking at. So in order to look at if the global signal changes across the lifespan, or the topography of the global signal changes across the lifespan, we had a large publicly available data set, NKI enhanced. We had 602 subjects, 6 through 85 years old. We had some standard pre-processing here, including ICA fix and a bandpass filter. And what we did was we did a general linear model in FSL where we took these whole brain voxel-wise global signal beta maps from each subject, and we ran two models. So the first model, we had linear age and frame-wise displacement as a nuisance covariate. And in model 2, we had linear age and quadratic age, along with frame-wise displacement again as a nuisance covariate. And we did cluster correction according to Eklund et al. And the results of the linear model showed that the thalamus and the right ventral insula had increased association with the global signal across the lifespan. That is, the relationship of the global signal to the bold signal in these particular areas increased, their association strength increased across the lifespan. We also found a lot of brain areas that had a negative linear lifespan relationship. So areas such as the medial prefrontal cortex, we had temporal areas, inferior frontal gyrus here, sensory motor areas, and subcortical areas such as the basal ganglia, all had a negative relationship with the global signal. That is, the global signal was more weakly associated with the bold signal in these brain areas as individuals got older, as they progressed across the lifespan. And finally, what was interesting was we found a positive quadratic lifespan effect where the global signal was most strongly related to these brain areas early on in life, and then it had a weaker association during the midlife period from around 40 to 60 years old. And then the association strength increased again towards the end of life. And this pattern was most prominent in this lateral frontal parietal network again here, or the central executive network. And then we also found that in subcortical areas such as the putamen. So in conclusion, we did find that global signal topography changes or shows linear and quadratic changes across the lifespan. And also, again, we found a linear increase, a linear decrease, and positive quadratic changes across the lifespan in distinct brain areas and brain systems. So one question is, are these specific spatial changes related to neural or vascular causes? And that we're not really sure about. So that's one thing we're going to look further into, perhaps using the NKI cardiac data they have. Because previous research has shown that distinct brain areas can have different contributions of cerebral blood flow and cerebral metabolic rate of the oxygen use. So perhaps this may explain what's going on. In any case, these results do have implications for developmental and lifespan fMRI studies. So essentially, I guess people should be aware that regressing out the global signal may differentially affect distinct brain areas according to what developmental stage you're looking at. So I guess the take-home message here is that if you regress out the global signal, you might want to be aware of the distinct brain areas that are being differentially influenced by that global signal regression, and perhaps adjust your results and interpretations accordingly. So thank you very much for listening to this talk. I'd like to acknowledge our collaborators and also our funding agencies. And thank you again.
Thank you for giving me the great opportunity to have this presentation. Today, I'm going to talk about a framework for linking the brain structure and functional dynamics. In neuroscience, there is a core assumption that the brain structure governs ongoing function, which means the brain function could be established according to the structure patterns of the brain. Many studies try to understand the underlying mechanisms of the correspondence between brain structure and function in terms of the brain connectivity. Structure connectivity represents the anatomical connections between the brain regions, and it can be inferred via diffusion MRI by approximating structural wiring in white matter through tractographic algorithms. And functional connectivity is the statistical relationships which can be calculated by the correlation of the time series between different brain regions. So here, the important question is how the brain structure and function are related in terms of a large-scale brain network. Previous studies tried to associate the brain structure and function using the large-scale connectivity information. For example, by estimating joint singular vectors from the structural and functional connectome data, or simulating the functional connectivity based on the structural connectome information. However, there is one thing we have to consider. Most of the previous structural function coupling studies were performed based on the static functional connectivity. The brain, however, is an inherently dynamic system where function and cognition involve time-varying network reconfiguration patterns. Especially, it remains unclear how the hardwired brain architecture supports functional flexibility that is at the core of the higher-order cognition. So now is the time to focus on the correspondence between the brain structure and function in terms of the dynamic functional connectivity. So in this study, what we want to explore is to construct a framework for linking the brain structure and functional dynamics. The brain structure was quantified using the manifold learning techniques. The manifold learning is a method of compressing high-dimensional data into a much simpler format by projecting the data into a low-dimensional representation. The main idea is to reduce our connectome data into a series of continuous dimensions, which represent the principal axis of the whole brain connectivity pattern. And I will refer this low-dimensional feature as a connectome manifold or gradient. Okay, then the functional dynamics were quantified using the hidden Markov model. It is a probabilistic framework that identifies time-varying brain states and associated connectivity profiles. A hidden Markov model assumes the underlying internal state that is not directly observable. Instead, the model tries to infer the hidden characteristic of the internal state. So from now on, I will introduce the study for linking the brain structure and function Using the diffusion MRI data, we first constructed the structural connectivity matrix. Then we applied the manifold learning to the connectivity matrix, and we selected the top two components, which showed almost 45% connecting variance. And I will refer these two manifolds as structure gradients 1 and 2. As in the figure, we can see each gradient represents different cortical axes. The first gradient differentiated the sensorimotor cortex from medial prefrontal ankle, and the second gradient was extended from the ventral to dorsal areas. The functional dynamics were quantified using the hidden Markov model. Based on the transition patterns between different brain states, we defined the two distinct clusters that showed similar transition patterns. And I will refer these clusters as metastates. These metastates show very interesting patterns. The first metastate seems like it has high activity in the sensorimotor areas, and the second metastate has strong activity in the default mode and the frontoparietal networks. So we can say that this model could infer the hierarchical organization of the brain. And now we're going to associate the brain structure and function using the estimated structural gradients and the transition patterns between the metastates. Here, we simply calculated Pearson's correlation to assess structure-function coupling. As you can see the figures, we could find strong relationship between the structural gradients and or the transition patterns, except for the transitions within the second metastate. So we can say that the brain structure can explain the low-level transitions, but it is not well suited to explaining the higher-order transitions. To explore why the brain structure couldn't explain the higher-order transitions, we compare the dynamic transitions based on the network communication mechanisms of the structure connecting data. If the network is organized in a very diffuse way, it will be quite difficult to transfer the information from the source node to the target node. In contrast, if the network is very well organized, the information will be traversed efficiently through the shortest path. To measure the network communication mechanisms, we calculated the mean first passage time and path length. They both represent the amount of the required cost to transfer the information between the two distinct nodes. So the high value means that it is difficult to transfer the information, and thus the network follows the diffusion mechanism. Interestingly, we found higher values for the low-level transitions, and the values were decreased for the high-order transitions. So we can say that the low-level functional states involve network diffusion mechanism, while the high-order transitions employ more routing mechanisms along the shortest path. So taken together, we found that the structure connecting manifolds can explain the dynamic transitions that involve low-level sensorimotor states, and the magnitude of the structure-function coupling becomes weaker if the transition is anchored to the high-order transmotor state. And we also found that these two dynamic states follow different network mechanisms. Okay, thank you all for listening to my talk, and thanks to all my collaborators and funding sources. And especially thanks to Human Connecting Project for providing the data. Thank you.
Great, thank you very much for joining my presentation today, and thanks to the organizers for giving me the opportunity to present our work. Now, as probably most of you know, the human brain is an astonishingly complex organ that comprises more than 80 billion neurons that form a network of densely interconnected neuronal populations. So from that, the notion of functional integration or brain connectivity seems to be important when trying to understand the organizing principles of our brains. And this is not only true for health, but also for disease. So various psychiatric and neurological disorders have been associated with global disconnectivity across the brain. Now, so far, human connectomics has mainly focused on structural and functional connectivity to study the brain. And those measures have indeed provided quite substantial insights into the organizing principles of our brains. However, they are essentially undirected measures, and in that sense, they are somewhat limited. What we rather want to have is measures of directed or effective connectivity. And one way to do that is dynamic causal modeling, which allows us to infer upon directionality. And the method has been used quite frequently, yet also comes with quite a number of substantial limitations. And one of them is that we are really restricted to very small networks that comprise maybe 8 to 10 regions. And the reason for that is that for larger networks, model inversion just becomes computationally infeasible. Now, that's not necessarily an issue if you have very clear hypotheses about the neuronal circuits that you want to target. However, often we are confronted with a situation that looks more like this, where we have global reorganization of neuronal activity, or here as measured with bold activity, throughout the cortex, here exemplarily shown for the resting state. In these cases, picking a couple of nodes is really challenging and doesn't adhere to the full complexity of the pattern. So in situations like that, what we really want to have is a model that can infer upon whole brain effective connectivity. And we have recently introduced regression DCM as a novel variant of DCM for fMRI that tries to move in that direction. Now let me give you a very brief glimpse of what the model does. We start off with a linear DCM in the time domain, which basically means we have directed interactions among neuronal populations and then also driving inputs that can change the neuronal activity in those neuronal populations. And then by using the Fourier transformation, we can bring this system into the frequency domain, and under additional reformulations, modifications, and prior specifications, we can turn this linear DCM into a Bayesian linear regression-like model in the frequency domain. And the important thing here is that these kind of models can be solved extremely efficiently. Now, we have also introduced sparsity constraints to the likelihood function of that model, which now allows us to basically start any whole brain effective connectivity analysis from a fully connected network and then pruning this network to the most essential connections given the data that we have measured. Now, for this approach, we have done quite a lot of simulations in order to demonstrate the phase validity of the model. And we have also done some applications to empirical data sets to show the practical utility of the approach. And I want to show you an example of the latter one. So here we have measured 25 healthy controls using a relatively simple visually guided hand movement paradigm, where you can then typically observe these classical activation patterns that are associated with motor actions. We then used the human brainotome atlas to parcellate the entire brain into 208 brain regions, and we extracted bold signal time series from each of those parcels of the atlas in every subject. So this left us with a DCM that comprised 208 brain regions, which covered the entire cerebrum and is an order of magnitude larger than what has typically been possible using DCMs. For this model, we now used in a first step the structural connectivity matrix that came with the human brainotome atlas and used that in order to inform the network architecture of our models. So this left us with almost 18,000 parameters that we had to estimate. And when we do that, we find average effective connectivity patterns that look like this. So here we have the A matrix of the DCMs, which tells you how strongly each and every brain region is connected to another brain region. And we have the C matrix here, which is the driving inputs, which tells us how strongly the visual stimulus activated or deactivated each and every brain region. And I don't want to go into too much detail here, but just to note that the patterns were quite plausible biologically, because we found very strong functional integration among motor-related regions in the pre-central gyrus and post-central gyrus, as well as other regions that were related to the task. For instance, visual regions, which more adhere to the visual pacing aspect of our paradigm, but also parietal regions and frontal regions, which then more adhere to the attentional aspects of our paradigm or the executive control aspects. Now, in the next step, we could make use of the fact that we had not only left-hand movements, but also right-hand movements in two separate sessions. So we could investigate the differential effect of the hand movement condition. And here I've shown all the connections that showed a significant differential effect of the hand movement condition, where green arrows indicate connections that were stronger during left-hand movements as compared to right-hand movements, and red arrows indicate the opposite. And these connections were multiple comparison corrected using the false discovery rate across all of those 18,000 connections that we had. And what you can see is that you find this beautiful mirror symmetric pattern, which is exactly in line with what you would assume. So connections among motor-related regions in the contralateral hemisphere seem to be enhanced during unilateral hand movements. The beautiful thing about this is that we find this pattern not only when we start from this structural connectome from the human brainotome atlas, but also when we start from a fully connected network and then use the sparsity constraints to prune this network. So this is an even more challenging situation, yet still we find this mirror symmetric pattern where regions or connections among motor-related regions in the contralateral hemisphere are enhanced during unilateral hand movements. Now, in the last bit of my talk, I want to give you a very brief glimpse of how we can now use this model and bring that to clinical applications. And for that, we looked at the data from the BSNP consortium, which is a very large multi-site study in the United States, where psychotic patients, first-degree relatives of those patients, and healthy controls were measured. And this left us with almost 1,000 participants. We now used our regression DCM approach to again fit whole brain models to the resting state in this case. And we then could investigate whether there are any significant group differences. And indeed, we found that patients differed significantly from first-degree relatives and from healthy controls in quite a substantial number of connections. And this was, again, multiple comparison corrected across all 18,000 connections. Now, interestingly, we did not find a significant difference between relatives and healthy controls, speaking to the fact that patients were significantly distinct from those two groups. And we found that differences not only manifested at the group level, but that we could use the whole brain effective connectivity patterns also to distinguish patients from relatives and patients from healthy controls at an individual level. And this was done using classification analyses, using a support vector machine, and tenfold cross-validation procedures. And we found that patients could be significantly distinguished from the other two groups by using permutation tests in order to generate a null distribution and then comparing our balanced accuracies against that null distribution. Now, obviously, this is just a first glimpse into how a clinical application of this model could look like. There's more work to be done, obviously. Now, if you find this approach interesting, RDCM is open source and can be downloaded as part of our tapas toolbox, which you find under the given link here. And with that, I would like to thank all my collaborators and the funding agencies and you for your attention. Thank you very much.